Sample size is one of the most critical aspects of medical research. But what exactly is it? Sample size refers to the number of participants or observations included in a study. It directly impacts how valid, reliable, and generalizable the study results are. A sample should be large enough to detect an effect if one exists, but not unnecessarily large. Several factors influence the determination of sample size in a study. First is study design. Whether it's observational, experimental, or a randomized controlled trial. The more complex the design, the more participants may be required. Population variability also matters. The higher the variability within a population, the larger the sample needed. The expected effect size is another key factor. A large effect may need a smaller sample, while small effects require more participants. Then there's the significance level, or alpha, typically set at 0.05. Power, or 1 minus beta, is also critical and is generally set at 80%, representing the probability of correctly rejecting the null hypothesis. Additionally, the type of outcome, whether categorical or continuous, and the attrition rates, which account for participants dropping out, can all influence sample size. Different types of studies also require unique sample size considerations. For example, cross-sectional studies often require large samples to be representative. Cohort studies base their size on the expected incidence of outcomes, while case control studies depend on exposure prevalence. Randomized controlled trials need to balance the detection of a treatment effect with ethical concerns. When calculating sample size, several statistical methods are available. Simple random sampling relies on known formulas based on expected effect size and variability. In cluster sampling, larger sizes are needed to account for intracluster correlations, while stratified sampling requires proportional samples for each group in the population. Software tools and calculators can simplify these calculations. G*Power is a popular free tool, while NCSS and PASS offer comprehensive features. Online calculators also provide quick solutions for basic designs. The impact of sample size on study outcomes cannot be overstated. An underpowered study may fail to detect real effects, leading to false negatives. On the other hand, an overpowered study might detect trivial, clinically irrelevant effects. Larger samples boost external validity, improving generalizability, while also enhancing internal validity by ensuring consistency and accuracy of conclusions. Ethical considerations are always paramount. It's crucial to minimize harm and unnecessary risks by not enrolling too many participants in overpowered studies. Informed consent must be obtained, with participants aware of the potential outcomes. Ethics committees play a key role in reviewing sample size decisions to ensure they are both scientifically and ethically sound. Challenges exist, of course. Recruitment can be difficult, especially for rare diseases or low-prevalence conditions. Funding constraints often limit sample size, impacting the power of a study. And participant dropout, or attrition, can reduce the effective sample size, affecting results. Some real-world examples demonstrate the importance of appropriate sample size. In landmark trials, such as cardiovascular drug and vaccine trials, accurate sample size calculations were vital to their success. Conversely, miscalculations have sometimes led to poor outcomes and even affected public health. In conclusion, sample size in medical research is a balancing act. Too small, and you risk missing important effects. Too large, and you may overburden the study. With advances in statistical methodologies and data collection, the future of sample size determination is likely to become even more refined and adaptive, helping to ensure ethical, efficient, and impactful medical research.